All right, so on to part 2 of your notes. Again, you're just paying attention to the stuff in green. That's what you need to have in your notes in order to get points for having participated in class. So once the pigments in the chloroplast have captured the light energy, it has to be converted into chemical energy. The chemical energy is temporarily stored in ATP and NADPH. During these reactions, oxygen is given off, which is why it's really great to have plants around you because you are getting oxygen from them. And in turn, you're giving them the carbon dioxide that you're breathing out. So they're using that carbon dioxide, which hurts us, and we're getting that oxygen, which they don't need. Photosystems are a cluster of chlorophyll and other pigment molecules. They harvest light energy for the light reactions of photosynthesis. There are two photosystems, photosystem 1 and photosystem 2. So whenever you're looking at this here, let me hide that. This is within a thylakoid. So right now we're in a chloroplast. We're looking at one of those tiny little discs. And you see that photosystem 2 is actually labeled first. And it's kind of crazy because they didn't know that photosystem 2 even existed. What happened is that whenever they were going through and looking at photosynthesis, they actually ended up starting right here in this area, and they found this little green guy here, and they said, oh, okay, well, this is photosystem 1. Great. So the electron's going to enter, there's going to be light energy, it's going to go to a primary electron acceptor, then through the electron transport chain, and it's done. Well, a little while later, they ended up finding out that there was yet another photosystem. So we actually start off with photosystem 2, go through an electron acceptor, a chain, and then it hits photosystem 1. And then it goes through another acceptor and a chain. So they found things a little bit backwards, which is why we have photosystem 2 in front of photosystem 1. So steps to convert light energy to chemical energy. Step 1, you're going to have light energy forces electrons to enter a high energy level. Whenever electrons see light, they get super excited and they start buzzing all around. So they're going to enter into photosystem 2. The excited electrons are going to leave the chlorophyll A molecules and are picked up by a primary electron acceptor. Right here, they're accepted in. Step 3, the primary electron acceptor is going to donate those electrons to the molecules inside the thylakoid. These molecules are known as the ETC, which is the electron transport chain, because they transfer electrons from one molecule to the next. Now, as these electrons are moving through, they're going to lose energy, so they're going to start to lag, and they're going to get a little tired, they're going to move more slowly. The energy is used to move protons in the form of H+, hydrogen ions, into the thylakoid. So far we've done step 1. Light energy came in, buzzed up our electrons. They moved to the acceptor. Then they moved to the chain. Now they're starting to get tired. And all while doing this, they're forcing protons away from them. Then they make it to photosystem number 1. They're going to get hit with light again, so we're going to buzz them up again, get them all excited. The electrons are going to move from 2 chlorophyll A molecules in photosystem 1 to yet another primary electron acceptor. This primary electron acceptor from photosystem 1 is going to then give those electrons to a different electron transport chain. The electrons from step 4 are brought to the stroma side of the thylakoid. Here, the electron is combined with a proton and NADP+, and this turns NADP+ into NADPH. In step 4, electrons from chlorophyll molecules in photosystem 2 are going to replace the electrons that leave the chlorophyll molecules in photosystem 1. If the replacement did not occur, the electron transport chain would stop and photosynthesis would not occur. The replacement electrons from photosystem 2 are provided by water molecules. An enzyme in the thylakoid is going to split water molecules into protons, electrons, and oxygen. So we have this formula here, H2O, or 2H2O plus yields 4 H pluses plus 4 electrons plus oxygen. And that is what we breathe in. The ATP is the main energy currency of cells, and chemiosmosis, from our vocab, is the synthesis of ATP through the movement of protons down the concentration gradient. Now recall that protons are produced by splitting water molecules inside of the thylakoid, and then they're going to be pumped from the stroma to the interior of the thylakoid. The concentration gradient of the protons is going to provide energy for ATP synthase. The synthase is going to add a phosphate to ADP, making it into ATP. So here we have ADP. We got this little proton. We're going to add that, no, not proton, phosphate. We're going to add this phosphate to this molecule here, making it into ATP. When we need energy, we're going to zap that phosphate off, and we get a lot of energy to do whatever activity we're trying to do. Through that electron transport chain, it's going to add that phosphate back on, and it's just a never-ending cycle there of making energy and creating it, or breaking it, making it, breaking it. The energy allowing ATP to be formed from ADP is from the movement of protons from inside the thylakoid of the stroma. So here's light. Photons are light. Come in, buzzes up those electrons. They go through the electron acceptor to the chain, and then they get buzzed up again whenever they see light in photosystem 1. At this point, they are pushing H+ ions into the thylakoid space, which is going to in turn push some back out, and you get energy. At the end of the light reactions, we are left with NADPH and ATP that will provide energy for the second set of reactions in photosynthesis. Again, I'm not going to play a video because I don't want to eat up too much of your time, but that is the end of section 1. Now, I realize that there's a lot of abbreviations in here, and your head's probably swimming like, oh my gosh, what is she trying to tell us? Don't worry so much about all the abbreviations. Just know the basic steps. We start at photosystem 2. Primary electron acceptor. Electron transport chain, photosystem 1. Primary electron acceptor, electron transport chain, energy. We have a couple review questions to go through, and I will do those in a separate video.